Welcome, everyone, and thank you for tuning into this Genetic Engineering and Biotechnology News webinar. It's entitled Use of a Knowledge-Based Human Protein Atlas for the Analysis of Gene Expression Patterns and Biomarker Discovery. The Human Protein Atlas project was established to allow for a systematic exploration of the human proteome using antibody-based proteomics. This is accomplished by combining the high-throughput generation of affinity purified antibodies with protein profiling in a wide range of tissues and cells assembled on tissue microarrays. Our webinar panelists are here to discuss how scientists can best benefit from tapping into and working with the Human Protein Atlas. Let's meet them. Dr. Fredrik Pontin is a professor of pathology at Uppsala University and clinical director of the Human Protein Atlas. Dr. Pontin is going to talk about gene expression analysis and biomarker discovery using the Human Protein Atlas. Dr. Henrik Verniers, Chief Scientific Director of Atlas Antibodies, is specifically going to focus on biomarker discovery with the Human Protein Atlas. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Fredrik Pontin will be our lead-off presenter, so I'm turning the program over to him. Fredrik?
Thank you, John. And I will start my talk over gene expression analysis and biomarker discovery using the Human Protein Atlas by going through a little bit of the history of this program, and then I'll give you an update of the current status and talk a little bit about where we are going and with respect to biomarker discovery and so on. So to begin with, then, the Human Protein Atlas project was launched some 10 years ago, and it was launched as a joint effort between the Royal Institute of Technology in Stockholm and the Uppsala University. And we had a very generous funding by the non-profit Knut and Alice Wallenberg Foundation, and today we are a little over 100 researchers that are financed by this grant in the core project. The vision behind this project was very timely. Just after the human genome was published in Nature and Science, 2001, we had a vision that we would generate validated protein probes towards all non-redundant proteins encoded by the human genome, and then to functionally explore these proteins. And the more specific goals can be divided up into three, and that was to generate and validate antibodies towards all human proteins. And when I say all human proteins, I'm meaning then a major isoform encoded by each protein coding locus of the genome. Secondly, we wanted to use these antibodies to create a map showing the pattern of protein expression in human cells and in human tissues and organs. And the third goal was then to utilize both these reagents, the antibodies, and also the protein expression data to try to identify and explore biomarkers of clinical relevance. To do this, we set up a multidisciplinary program where we identified and defined different modules and units, starting with an upstream bioinformatics module, and then continuing with molecular biology, a protein factory. We then outsourced the antibody production, and then an immunotechnology unit where we then could purify our antibodies. Our purified antibodies are then tested in various assays where we use antibodies, and all the data is then finally released and made publicly available on the Human Protein Atlas portal. I will very briefly describe some of the upstream parts, and I'll focus mostly on how we use the antibodies. But this slide then shows our antigens and how we generate our antigens. So we use bioinformatics and define protein fragments of approximately 100 amino acids length. And by using then a blast function, we can then look for the regions of this protein which have the lowest homology to all other proteins encoded by the genome. this allows us then to select a protein fragment, which we call PRESTs, protein epitope signature tags, which has as low homology as possible, enabling us to use this as an immunogen to get antibodies, which then hopefully show a high level of specificity in various assays. After having generated or after having defined the region for PREST, we then make primers, and then we use RT-PCR to fish in a pool of human RNAs and clone that fragment then into vectors that then can be transformed into E. coli, where the recombinant protein is then produced, and after production then purified. We make rabbit polyclonal sera, which is outsourced, and after getting the sera back, we then purify the sera to make affinity purified antibodies. And these antibodies, when one does epitope mapping, usually shows about 3 immunogen epitopes. So one could say that they are rather oligoclonal in their nature. These antibodies, when purified and tested, are then used for protein expression profiling, and we do that on several different levels, using light microscopy and immunohistochemistry to look at protein expression patterns in organs, tissues, and also cells to some extent. And then we use immunofluorescence and confocal microscopy to look how the proteins are expressed on the subcellular level. To be able to also get a wide coverage of tissues and cells, we use tissue and cell microarrays. And tissue microarrays was also very timely described at the end of the 90s at NCI by the group of Kallionemi et al. And I think this slide shows you the beauty of using tissue microarrays. Instead of handling hundreds of different tissue blocks, one can then drill out the representative core of tissue, assemble it into a tissue array, and then from that tissue array one can stain some 200 to 300 antibodies. In the Protein Atlas project, we have focused on normal tissues, and we have then collected normal tissues from 144 different individuals, representing 45 different major human tissue types. A further focus was to also explore cancer, and here we have 216 different individual tumors representing the 20 most common forms of human cancer. And in addition to that, we have a cell array with some 50 cell lines that is also probed with each antibody that we have approved. So altogether, every antibody that we have approved is then stained on 708 different spots of either not normal human tissue, cancer tissue, or cell lines. After having immunohistochemically stained these tissue and cell microarrays, they are scanned to produce high-resolution digital images, and these images are then used to annotate the protein expression patterns. For cells, we use image analysis-based algorithms to measure the intensity of staining and the pattern of staining. But for tissues, there are really no such developed algorithms that can distinguish a cancer cell from a normal cell, etc. So here we rely on pathologists to do the evaluation of immunohistochemical outcome. And we have a fruitful collaboration with a great team of pathologists in Mumbai, India, headed by Dr. Sanjay Navani, who then, together with his team, looks at the microscopical images and annotates where the protein is expressed and where it is not expressed. And this image is just to display the enormous amount of data that is withheld in tissues stained for a certain protein. And I think it really shows you how immunohistochemistry, which is not a quantitative method, but an extremely useful method to look at spatial expression patterns, where one can see what cell type or which cell types or which structures in a complex tissue do actually express the protein of interest. In addition then to protein profiling using bright field and tissues, another very important aspect is then where in the cell is a protein expressed. And here we use then immunofluorescence and confocal microscopy, and we then test different cell lines, and based on their level of transcription of a given gene, we then select that cell line to probe with our antibodies. And of course, the advantage of using immunofluorescence is that one can use different color channels and mark the different compartments within the cell. And we use the blue channel for Doppler staining of the nucleus, and then we have the red channel to outline the cell with the cytoskeleton, and then we have a yellow channel for the endoplasmic reticulum. And then we can come on with our antibody in green, and you see some examples in the background of the current slide. All this data, both from the normal tissues, from the cancer tissues, from the cell lines, and from the immunofluorescently stained cell lines, is then made publicly available on the Human Protein Atlas portal. And I'll come back to the Human Protein Atlas portal at the end of my talk. I first want to talk a little bit about the validation of protein expression, and this is key for using antibodies, is to validate that the antibody actually binds the intended target. And we spend a lot of time on trying to develop validation protocols, both basic validation. We do gene sequencing of our pressed clones, and we do mass spec on our purified recombinant protein to see that we immunize with the correct immunogen. And then we also perform protein arrays to see that our antibody specifically binds to the protein fragment that we have used as an immunogen. All those antibodies which then pass the basic validation, we are approved, and they are tested then on a standardized Western blot, where we then look at the lysates from 2 cell lines, human plasma, and then 2 different human tissue types. We also run them in immunofluorescence, and here we can compare the outcome of immunofluorescence with literature and bioinformatic predictions of where in the cell the protein should be expressed. And the same with immunohistochemistry, where we can search for literature and also to some extent use bioinformatic prediction. But the difficult part is, of course, to validate the pattern of protein expression in tissue. And remember that for about 30% of all our genes, there is virtually nothing known about where the protein is expressed at all. So there is no blueprint to compare your results with. And I'll come back to how we're trying to address this difficulty. Just first a basic slide and a reminder to everyone about antibodies and specificity. Antibodies have a specific binding, and one talks about specific binding to the intended target, which is for a good antibody in the nanomolar range. But one should also remember that all antibodies bind specifically to other proteins, although in a millimolar range, which is quite different than from the unspecific binding, where the antibody binding site of the antibody is not involved, which is more of a noise that one can have in various antibody assays. So this means that your specificity of the antibody is both dependent on the affinity to the intended target, but also the affinity to the off-targets. And of course, it's context dependent, so it also depends on the concentration of targets and off-targets. And I think this is a very important point to remember when one doesn't have a blueprint, again, to compare with. That one can have specific off-target binding, and one has to then be careful how to interpret signals from immunohistochemistry. So what can one do when one doesn't know what to expect? Well, one can use paired antibodies, and we try as much as we can to then produce at least 2 antibodies towards one protein, and antibodies then that bind non-overlapping epitopes of the same protein, and one can use these antibodies for double stainings with different chromogens. One can use these antibodies for staining consecutive sections and compare the results, or one could use both antibodies in assays which require 2 binding events to produce a signal, such as the proximity ligation assay. One can also compare the result of antibody binding with the underlying expression of specific transcript. There are ongoing efforts to multiplex this using padlock probes, but one can also just look at use RNA sequencing to quantify the number of transcripts derived from various tissues or cell lines and compare them with the antibody-based immunohistochemical staining. And I'll go through an effort where we have worked with then comparing transcriptomics and then antibody-based profiling using immunohistochemistry. So the last 2 years we have spent a lot of effort to collect well-defined normal human tissues from the Uppsala Biobank, altogether over 100 different samples representing 27 different tissue types as outlined on the slide. We have then done RNA extractions from cryosections, and from these we have done sequencing to then retrieve quantitative mRNA data from our 20,000 genes. And this was recently published at the end of last year. The quantitative data from RNA has allowed us then to classify the human protein coding genes, and we find then that a little less than half of all our genes, 46%, are expressed in all the different tissues, and these can be considered as being housekeeping genes that are expressed in any cell type independent of function or morphology. These are depicted in red in this pie chart that you see. 9% of all the genes we didn't find any transcript of, and these could either be pseudogenes or else genes that are only expressed in very specialized tissues that we did not have among these 27 tissues, or perhaps genes that are expressed during development and turned off in cells of adult tissues. About 15% or 17% of our genes can in some respect be categorized as being tissue enhanced or tissue enriched, showing an enriched specific expression in one tissue type. And of course, depending on cutoffs and definitions, one can tweak these numbers a little bit. But about 15 to 20% of our genes make up for the tissue specificity and the differences between different organs and tissues. And we have here divided them into genes that are highly tissue enriched, meaning that they are expressed at least 50 times higher in one tissue than in all the other 26 tissue types, or moderately tissue enriched, being expressed at least 5 times higher levels than in any other tissue type. We also have a group which we call group enriched, where a gene is then specifically expressed in only 2, 3, or 4 different other tissue types. The next slide shows you a cytoscape plot where then all the different tissue types that we have are in gray circles, and then all the high and moderately enriched genes are in light blue circles. And as you can see from this slide, the tissue type that has the most tissue enriched genes is testis, with 1148 genes that are then high or moderately tissue enriched in testis. Testis is then followed by the brain and liver, which also have quite a few tissue enriched genes. The dark blue circles of this cytoscape plot then shows you the genes which have shared expression between 2 or 3 different tissue types, those that I previously mentioned as being group enriched tissues. And as you can see from this cytoscape plot, you can see that skin and esophagus, for instance, has over 70 genes that are only expressed in esophagus and skin, and these are then the genes which are involved in squamous differentiation and so on. You can also see that the organs rich in immune cells, such as appendix, lymph node, and spleen, share several genes, or the tissues from the gastrointestinal tract share a number of genes. So there's quite a lot of information in this cytoscape plot, and of course, very much more information when one goes to the list of genes and looks what proteins do they encode for. The next slide shows you a couple of examples of then these tissue-specific genes and how the corresponding protein is expressed. In the top level, you can see kidney-specific proteins expressed in the glomerulus to the left, and then in proximal tubules in the center and distal tubules to the right. In the middle panels, you can see liver-specific proteins and the pattern of expression that they show from the portal zone to the central vein. And at the bottom of the slide, you can see examples from the testis with testis-specific proteins being expressed in various stages during differentiation. And I'll give you one example from one of these different tissue types or organ types, and that'll be testis. So here we had normal testis from 7 different individuals, and when we then looked at our mRNA data, we could see that about 75% of all the human genes, 15,224 genes, were expressed in the testis, at least at a level of 1 FPKM. At the bottom of this slide, you can see also that these individual samples showed a very high correlation, varying from 0.92 to 0.99 when we tested the different samples towards each other. When comparing to other tissue types, you can see the correlation down to the right of this slide, and where testis showed the highest correlation to ovary and the lowest level of correlation to the liver. The next slide then shows you the categories. If we look at now only at the genes expressed in testis, again, of course, the housekeeping genes are the same housekeeping genes in any tissue, 46%. In the testis, 23% could not be detected. And then you can see that 2% of the genes were highly tissue-enriched and 4% were moderately tissue-enriched in the testis. This corresponds to 384 genes, which are then specifically expressed at least 50 times higher in testis than in any other tissue, and 760 genes which are expressed at least 5 times higher in testis compared to other tissue types. The pie chart to the right shows you then the proportion of mRNA molecules in these categories. That is, if we look at the housekeeping genes, which then make up for 46% of the genes expressed in testis, they contribute with almost 80% of all the mRNAs in the samples taken from testis. And these highly testis-enriched genes contribute with 6% of the mRNA pool. Again, a cytoscape plot now having testis in the center with its 1124 high and moderately tissue-enriched genes, and then you see in the cytoscape plot the shared gene expression with other organs. And you can note here that testis shares gene expression mainly with the brain and with the lung. The shared genes with the lung are dominated by genes then that are expressed in the cilia of respiratory epithelia, but also expressed then in the mature sperm and genes involved in sperm motility and so on. These 1124 high and moderately testis-enriched genes, we went back to the literature and we found then that 62% of these genes were not previously characterized with respect to function or with respect to where they were expressed, in what cell type in the testis they were expressed in. If we took the cells which were expressed in testis and where localization had previously been assigned, which is the pie chart to the right, you can then see that a majority of these testis-specific genes were expressed in mature sperm or spermatids, and very few known genes that are testis-specific were expressed in early stages of testis development. And I'll come back to that a little later. This slide then shows what we can do with using immunohistochemistry. So here I've chosen to show some examples of then only highly testis-enriched genes. These are now the genes which are specifically expressed in testis and how they are distributed in various stages of spermatogenesis. The top panel then shows you those proteins which are expressed in the more stem cell near population in the seminiferous ducts, the spermatogonia, placed at the bottom or the periphery of where spermatogenesis occurs. And you can see to the left are some fairly well-known or characterized genes, but to the right there are genes which are totally unknown and previously uncharacterized. On the second panel from the top, you can see then genes which are expressed in the spermatocytes, and further down genes which are expressed in the spermatids. And at the bottom row, you see examples of genes showing protein expression in mature sperm. And also here some well-known genes to the left, proteome 2, while on the right hand of the slide you can see previously totally unknown genes encoded by open reading frames. If we then use our protein immunohistochemistry-based data and look then at this long list of highly testis-enriched genes, one can then find that several of the testis-specific genes are actually expressed in spermatogonia, as shown in the pie chart in the turquoise color. So, I think one can make some general conclusions from this example and from the effort of using RNA sequencing. I think that using deep sequencing and you start off with microscopically well-defined and controlled tissues, and you combine that with antibody-based profiling using the protein atlas, one can get a very detailed map showing the actual gene expression in the human body. I also think that quantitative transcript data provides a very nice basis to compare with antibody-based protein expression data, and thus functions as an additional validation for using antibodies to look at gene expression. And I'm convinced that combining transcriptomics and antibody-based profiling, if one then includes also disease tissue, has the potential to provide important knowledge regarding pathophysiology and allowing to link gene expression patterns to phenotypes. At the end of my talk, I will just briefly tell you a little bit about the current version of the Human Protein Atlas, version 12, which was released in the beginning of December 2013. What is new about version 12 is that we now base our gene expression data on both transcriptomics, the quantitative RNA sequencing data I just talked about, and also the protein expression data based on immunohistochemistry and immunofluorescence. It's a similar basic structure as the previous versions of the Human Protein Atlas, but we have subdivided the atlas into 4 major parts, depending on that the purpose of each part is quite different. So the normal tissue atlas is for tissue distribution and to show a map of where our genes are expressed. The subcellular atlas for subcellular localization of the proteome, and then for the cancer atlas, we will more focus on differentially expressed genes in cancer, and the cell line atlas will be more of a toolbox for researchers to find the right cell line when they want to perform their experiments using different genes or proteins. So, if the next slide then shows you an example of what the new protein atlas looks like. If you search for a gene, I have 2 examples here. On the left side, a housekeeping gene, showing then the RNA data to the left for each tissue type and the protein data to the right for each tissue type. The right half of this slide shows you a tissue-specific gene, one of these testis-enriched genes, and you can see the RNA data to the left and the protein data to the right as different color-coded bars. We also, for each gene, have selected 6 prototypical tissue types, which we show then the immunohistochemical image for on the first page. If you then click on one of these tissues, say that you click to the left then on heart and want to look at this housekeeping gene in more detail of the heart, you can then see the antibody data from, in this case, 4 different antibodies towards this malactohydrogenase 2 protein that we have stained for. You see smaller images of the heart muscle on the top. At the bottom of the page, you can see then the raw data from the RNA sequencing experiment, where you, to the left, see H&E stained frozen tissue sections, and then you see the actual Manhattan plots with a number of reads for the different parts of the gene, covering the different exons of different splice variants and so on. To the right, you see exactly the same thing, but for this tissue-specific gene, proteome 2, where you see that we have 2 antibodies against proteome 2, and you see the staining of different samples of testis, and then on the bottom, the Manhattan plots. So we still have the full high-resolution images, and the next slide shows you then on the protein side with the immunochemical staining for proteome 2 in the high magnification image of normal testis, and here you see the text box to the left where you get some of the details where the sample comes from, the age and gender of the patient and what he was operated for, etc. And then you can go into the picture and look for yourself for the details of where then the protein is expressed in the tissue. Lastly, we also show you the source of our mRNA pool from testis, and where you can find, in this case, 7 different examples of testis, and you can also go into this H&E stained frozen section and look at what cells actually contribute to the mRNA pool that we have our transcriptomic data from. So I'll just end up with one slide, then again showing you the first page of the Human Protein Atlas and the 4 different sub-atlases, and I hope you enjoy using the Human Protein Atlas as much as I do. Over to you, John, and thanks.
Frederick, thank you for an in-depth overview of the Human Protein Atlas. That was an excellent and wide-ranging introduction to a number of topics that we're going to continue to focus on today. If you're just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the webinar presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Henrik Bjernius, is ready to begin his presentation. So, Henrik, the floor is all yours.
Thank you, John. Hi, and welcome to the webinar. I'm Henrik Bjernius, CSO of the Swedish research antibody provider Atlas Antibodies. Today, we will discuss 3 things. First, what is a biomarker, and why do we need them, and how can biomarkers contribute to overall patient management? Secondly, how can you as a researcher take advantage of the Human Protein Atlas to do biomarker discovery on a proteome-wide scale? And finally, a brief presentation highlighting some of Atlas Antibodies' proprietary oncology biomarkers discovered through the Human Protein Atlas. But first, a few words about Atlas Antibodies, who we are and what we do. So Atlas Antibodies was established in 2006 as a spin-out from the Swedish Human Protein Atlas project. Our mission is quite simple. It's to provide you and other researchers worldwide with highly characterized primary antibodies to all human protein coding genes. Through our AAA polyclonals, we are proud to offer you the world's most advanced line of polyclonal research antibodies. These are developed by researchers for researchers and offer you advanced levels of specificity, reliability, and versatility in your applications. Today, we have more than 17,000 AAA polyclonals available off the shelf, covering 75% of the human protein coding genes. To learn more about us and the AAA polyclonals, please visit us at atlasantibodies.com. My first topic of today will be what is a biomarker and why are biomarkers important and how can they be used in various aspects of patient management. Every year, more than 13 million people around the world are diagnosed with cancer. There are close to 8 million cancer-related deaths. And today, the microscopic evaluation of tissue and tumor specimens by trained pathologists remains the gold standard to establish a firm diagnosis of cancer. However, this traditional pathology-based method and the prognostic factors such as stage, meaning how spread the cancer is, and the grade, assessing the aggressive potential of the tumor, are frequently insufficient tools for a correct estimation of individual patients' prognosis. And therefore, the analysis of protein expression in cells from a tumor tissue often provides important additional information to the responsible clinician. Immunohistochemistry, or IHC, using protein-specific antibodies, provides a tool to detect the presence, the abundance, and the localization of specific proteins. And with only a limited set of protein biomarkers available today, there exists a clear and unmet clinical need to identify novel sets of biomarkers. The word biomarker is a widely used term, and biomarkers can be defined in many different ways. One definition, according to the Food and Drug Administration, FDA, is that a biomarker can be defined as a factor that can be objectively and precisely measured and that reflects a physiological or pathological process or a therapeutic response. Biomarkers could be anything from observable characteristics such as blood pressure, pulse, or body mass index down to quantifiable changes in the genome, transcriptome or proteome of the individual patient. Biomarkers are frequently categorized into either diagnostic, prognostic, or treatment predictive. Diagnostic biomarkers show a highly specific expression pattern and can be used for detection of specific disease. They can be used for analysis of blood or other body fluids for early diagnosis or for tissue-based differential diagnostics. The prognostic biomarkers show a differentiated expression pattern that correlates in some way with the patient outcome. They are used to stratify, that is, to divide the patients into high versus low risk groups. This information could be used as a guide for patient information and monitoring. In the third and final group, we find the treatment predictive biomarkers, where the expression correlates with the pharmacological response to a specific therapeutic intervention. Predictive biomarkers are used to stratify patients into responders versus non-responders and offers the responsible clinician a guide for treatment selection. In the following slide, I try to summarize the various potential contributions of biomarkers to patient management with a figure from a publication by Brennan et al. It is widely believed that the discovery, the testing, and the validation of clinically relevant biomarkers will allow more personalized therapeutic intervention. As shown in this figure, biomarkers can play key roles in various areas of the patient management, from early screening programs through the diagnosis to monitoring of disease progression and recurrence. In addition to allowing a more individualized treatment, the adoption of novel biomarkers promise to significantly reduce public healthcare costs and to also improve the overall drug development process. In the second part of my talk, I will focus on biomarker discovery and how to use the Human Protein Atlas as a tool to identify novel cancer biomarker based on immunohistological stainings of human tissues and cells. The Human Protein Atlas, found at proteinatlas.org, is a unique world-leading effort to perform antibody-based immunohistological profiling of human protein expression in a multitude of tissues and cells. This comprehensive and open database offers researchers worldwide a platform to perform basic research on global gene expression, but also targeted discovery and validation of protein expression patterns of clinical relevance. With the most recent data release in December 2013, the Human Protein Atlas now offers transcriptome and proteome data to 82% of the protein-coding genes. The polyclonal research antibodies developed by this leading Swedish initiative, led by Professor Mattias Julen, are marketed by Atlas Antibodies under the brand name of AAA polyclonals. And at this stage, I would like to reiterate some of the points made by Professor Pontén during his presentation. The AAA polyclonals developed by the Human Protein Atlas team are used for immunohistological staining of an amazing amount of human tissues and cells that all have been carefully collected, validated, and used to construct a number of tissue microarrays. These arrays contain normal tissue samples from 144 individuals from 48 different tissue types. You have 216 tumor samples corresponding to 20 of the most frequently occurring cancer types. And finally, some 49 different cell lines and clinical cell samples. All these stained TMAs are digitalized. They are distributed over the web through a web-based annotation interface to trained pathologists who manually annotate each individual tissue core in terms of, for example, staining intensity, the fraction of stained cells, and the protein distribution. The cell TMAs are analyzed using an automated image analysis software. And all the annotations and the underlying images are freely accessible on the Human Protein Atlas. The Protein Atlas currently contains protein expression profiles for 16,600 genes based on some 22,000 antibodies. Each of these profiles contains manually annotated immunohistochemistry data from more than 700 tissue samples. At Atlas Antibodies, we perform advanced searches in the Human Protein Atlas for discovery of potential biomarkers of clinical relevance. This initial discovery aims primarily at identification of cell and tissue type specific or differentiated protein expression patterns. This is a prerequisite for finding biomarkers of potential diagnostic or prognostic value. The identified targets resulting from this initial screen is subsequently validated in carefully collected and dedicated independent sample cohorts that can be used to address specific clinical questions. At Atlas Antibodies, we have established platforms for anatomical pathology screenings based on these tissue microarrays. We have ongoing screening of serum and blood using AAA antibody bead arrays that allows multiplex screening of a large number of samples. And as a foundation for both of these discovery platforms, we have the underlying data of the Human Protein Atlas and our AAA polyclonals. In the third and final part of my presentation, I will share some selected examples of Atlas Antibodies' proprietary biomarkers discovered through the Human Protein Atlas using the AAA polyclonal. Approximately 5% of all cancers are first found as a metastasis from an unknown primary origin. This is also known as cancer of unknown primary. And in these cases, and when the clinical and morphological data are not in agreement, IHC provides a useful tool to narrow down the range of diagnostic alternatives and to establish the most likely site for the primary tumor. The Human Protein Atlas provides a useful tool to identify protein signatures to accurately distinguish tumors of different types and origins from each other. These can then be used for differential diagnostics of metastatic carcinoma of unknown primary origin. And in the present slide, I show a figure from a very recent publication by Gabriela Gremel, where they, in a systematic way, investigated the diagnostic power of 27 antibodies, all commonly used for clinical differential diagnostics, by looking at immunohistochemical stainings of more than 940 different tumor samples. Some of the antibodies, like PSA and thyroglobulin, showed excellent specificity and sensitivity, but the majority showed a lower degree of sensitivity and specificity for the defined cancer types. This figure illustrates in a good way both the benefits of immunohistochemistry, but also the unmet need for novel biomarkers to improve differential diagnostics of cancer. The special AT-rich sequence binding protein, SATB2, was recently identified by the Human Protein Atlas as a potential diagnostic biomarker for CRC. In an immunohistochemical analysis, SATB2 show a very specific nuclear immunoreactivity in cells of the lower GI tract, with only a subset of other tissues and cells being positive. The SATB2 expression has been analyzed in more than 1,900 colorectal cancer patients, coming from 1,700 primary and 200 metastases. And these come from 9 different independent sources. In addition, 8 other cohorts from unrelated cancers have also been analyzed, like prostate cancer, breast cancer, ovarian, and gastric cancers. In short, some 85% of all CRCs showed nuclear expression of SATB2, and this expression was also maintained in metastasizing colorectal cancer. And using the combination of SATB2 and CK20, more than 95% of all CRCs could be accurately identified. Also, Also, the role of SAT-B2 as a diagnostic complement to CK20, CDX2, and other known markers has also been determined in a pathology-based prospective study at the Uppsala University Hospital, where all clinical cases stained for CK20 was also analyzed for SAT-B2 expression. The next example is the RNA binding motif protein 3, RBM3. This is an RNA and DNA binding protein with an only partly known biological function. We do know that it's a stress-induced protein. It's one of the earliest genes to be induced upon cold shock, low oxygen tension, and other kinds of cellular stress. The potential role of RBM3 as an oncology biomarker was first discovered through the annotation of normal and cancer tissues in the Human Protein Atlas, where it also showed a highly differentiated expression pattern in various cancers. In the bottom part of the current slide, you can see representative IHC staining of RBM3 in urothelial cancers ranging from no expression to the left to high nuclear RBM3 expression to the far right. In the following slides, I will give you a few examples related to the role of RBM3 as a prognostic and treatment predictive biomarker. For a more comprehensive overview, please go to atlasantibodies.com/research/oncology biomarkers. RBM3 expression was first investigated in breast cancer using the corresponding AAA polyclonal. In subsequent studies related to CRC, we also developed and validated an anti-RBM3 mouse monoclonal, AMAB90655. These two antibodies were characterized in parallel through siRNA knockdown in transfected SW480 cells using untransfected cells as positive controls. Here, you can see the knockdown of RBM3 in Western blots, that's figure B, and immunocytochemistry in figure C, using the two different antibodies. We have also compared the two antibodies on consecutive tissue sections with near-perfect correlation. And in all subsequent studies, we have used the AMAB mouse monoclonal for RBM3 analysis. We have shown that low nuclear expression of RBM3 is associated with poor prognosis in several different cancer forms, like breast, ovarian, colorectal, prostate, and malignant melanoma. In a recent study by Carolina Boman and co-workers, which is summarized in the present slide, the prognostic impact of RBM3 expression in urinary bladder cancer was investigated. Some 20% of patients with urothelial carcinoma first present with muscle-invasive disease. However, for the majority of patients, the tumor does not invade muscle at diagnosis. And these early-stage tumors represent a real clinical challenge, which is subject to much controversy due to their highly unpredictable nature and potential for recurrence and progression into muscle-invasive disease. The real clinical challenge is here to identify these very early-stage, high-risk cases up front in order to offer them cystectomy as the primary treatment option. We have looked at the expression of RBM3 in prospectively collected material from the Uppsala University Hospital containing patients with newly diagnosed urothelial cancer. The results showed a significant association between reduced RBM3 expression and more advanced tumor stage in high-grade tumors. To the left, you can see the impact of RBM3 expression on 5-year overall survival in the complete cohort, with the negative RBM3 expression shown in red. And to the right, we have looked at overall survival in early, that's TA and T1 patients, again with the same trend. To summarize, RBM3 may be a valuable prognostic biomarker in urothelial cancer and a useful tool for improved treatment stratification of patients suffering from early-stage disease. We have also investigated the potential role of RBM3 as a predictor of response to platinum-based treatment. Although molecular biomarkers to determine the effectiveness of targeted cancer therapies have been widely adopted in colorectal cancer, those predicting chemotherapy sensitivity, still remain very limited and poorly defined. In a study by Åsa Elén, the RBM3 expression was examined in vitro by ICC using the cisplatin-sensitive ovarian cancer cell line A2780 and its cisplatin-resistant derivative A2780-CYP70. The results shown here to the left show a reduced RBM3 expression in the cisplatin-resistant cell line. And this potential relationship between RBM3 expression and cisplatin response in vitro was also examined using siRNA-targeted knockdown of RBM3 in A2780 cells, where it was shown that knockdown of RBM3 resulted in reduced sensitivity to cisplatin and demonstrated by cell viability assays shown to the image to the right. Judging from these initial data, RBM3 might be a predictor of response to platinum-based treatments. In subsequent studies, we have investigated the correlation between high levels of RBM3 and response to platinum-based treatment in stage 3 and 4 colorectal cancer. In this, although limited material, all patients with high RBM3 expression and that received adjuvant treatment survived until the end of the study period. We have also other ongoing studies to investigate this relationship further, both in vitro and in vivo. Finally, I would like to talk about polycalyxin-like 1, or PODXL. This is a single-pass transmembrane protein belonging to the CD34 family. The expression of PODXL is linked to cellular anti-adhesive properties, inhibiting cell-cell interactions through different charge repulsive effects. PODXL is normally expressed in hematopoietic progenitor cells, in vascular endothelial cells, and as shown in the image to the right, in glomerular polycytes, where it plays an important role in maintaining filtration pathways. Together with Karin Gistrom, the Atlas Antibodies R&D team have investigated the potential role of PODXL as a biomarker for poor prognosis in colorectal cancer. The overexpression of PODXL has previously been associated with more aggressive tumor phenotypes and adverse outcome in several cancers. The mechanism behind this association is not fully understood. However, it is known that PODXL plays an important role in a process known as EMT, epithelial mesenchymal transition. This process is an important part in the initiation of metastasis for cancer progression. The PODXL expression has been analyzed by immunohistochemistry in more than 1,000 colorectal tumor samples, originating from 3 independent sources. PODXL typically show a cytoplasmic expression with a membranous accentuation in selected cancers. Here, typical sample stainings are shown. From left to right, you have colorectal tumor with negative tumor cells and positive stromal vessels, serving as an effective internal control. You have moderate cytoplasmic staining without membranous staining, and finally, to the right, a strong membranous staining in a subset of tumor cells. Again, the AAA polyclonal and the mouse monoclonal anti-PODXL antibodies were carefully validated and used to stain consecutive tissue sections of the same cohort. And while the two antibodies correlated 100% in terms of identifying the patient showing strong membranous expression of PODXL, the monoclonal antibodies showed a somewhat cleaner staining with less cytoplasmic expression than obtained with the corresponding AAA polyclonal. The PODXL expression was initially investigated in CRC samples from the Malmö Diet and Cancer Study. In total, some 557 out of incident 626 CRC cases were available for TMA construction. In this study, there was a strong correlation between membranous PODXL expression and established clinical pathological parameters. As shown in this slide, these findings have also been validated in 2 additional cohorts containing more than 600 additional CRC samples. In Kaplan-Meier analysis, the membranous polycell expression was shown to be an independent marker for negative outcome, both when looking at 5-year overall survival and disease-free survival shown to the left and right, respectively. This was true both in uni and multivariate analysis when adjusting for TNM score, presence of vascular invasion, and differentiation grade. More than 1.2 million people are diagnosed with CRC every year, and even though mortality is progressively declining, it still remains the second most common cancer-related cause of death in the Western world. Early detection and adequate surgical removal and the optimal use of adjuvant treatment are critical to the clinical outcome. And in this study, we also looked at the impact of PODIC cell expression on survival in relation to adjuvant treatment in more than 100 curatively resected stage 3 patients. And as you can see on this slide, patients with membranous PODIC cell expression who were treated with adjuvant chemotherapy, that's the purple line, had a similar overall and colorectal cancer-specific survival as patients with low PODIC cell expression, the blue and green lines, respectively. On the other hand, untreated patients with PODIC cell high tumors had a significantly shorter overall survival and colorectal cancer-specific survival, as shown by the yellow lines. For the stage 2 patients, we could see a clear association between PODIC cell expression and overall survival, but the number of patients who had received adjuvant treatment were too few in this group for a meaningful statistical analysis. We have also looked at the prognostic value of PODIC cell expression in urothelial carcinoma, using the same cohorts as for the RBM3 studies. Again, as shown on this slide, there was a very clear association between PODIC cell high patients and reduced overall survival in both materials. So, for patients diagnosed with early-stage tumors, radical cystectomy should indeed be considered as a primary treatment option when found to be PODIC cell high. So, the take-home message from this webinar is that traditional methods for establishing a firm diagnosis of cancer are often insufficient to provide a correct estimation of patient prognosis. So, there is a need for new and better biomarkers. Improved biomarkers will advance cancer therapy. They will lead to more individualized treatment, improved drug development processes, and also savings in public healthcare costs. The Human Protein Atlas is a unique, world-leading antibody-based effort providing researchers with tools to do genome-wide expression studies as well as targeted biomarker discovery programs. At Atlas Antibodies, we are proud to offer you these AAA polyclonals developed by researchers for researchers to more than 75% of the human protein coding genes. I have also given you a few selected examples of Atlas Antibodies' proprietary biomarkers like RBM3, PODIC cell, and ZB2, all discovered using the combination of the Human Protein Atlas and AAA polyclonals. And with that, I would like to thank you all for listening to this webinar, and I look forward to taking some questions. For more information about Atlas Antibodies, the biomarkers presented here today, or the AAA polyclonals, please visit us at atlasantibodies.com, and you can also follow us on LinkedIn. And now, back to you, John.
And thank you, Henrik, for that insightful discussion on biomarker discovery using the Human Protein Atlas. Based on your presentation and Frederick's, I really think our webinar attendees now have a much greater appreciation of what the Human Protein Atlas can offer them in terms of research possibilities. Indeed, it looks like our audience appreciated your presentations because they have sent in some very interesting questions. But before we begin our Q&A session, I'd like to ask you to disable your pop-up blockers. A short survey on this webinar will appear in a moment, and we would very much appreciate your feedback on the webinar. So, without further ado, let's take the first question from our audience. Okay, Frederick, we have a question, and the first one is... It's a question here. So, any research on genetic variants of tissue-specific expression of proteins, captured, and population distribution in the Human Protein Atlas?
The short answer, excuse me?
Yeah, go ahead.
Okay, the short answer to that is no, and I'll just take the opportunity to clarify a bit. We get all our samples from surgical specimens, and we have the ethical permit to do this protein profiling dependent on that we use anonymized patient material. That also means that we have no sequencing data. We don't know if one of our normal breasts or breast cancers have a BRCA mutation and so on. So the answer is no, but this is of course something that one would like to do in the future, and I'm sure that with all the ongoing exome sequencing and whole genome sequencing, that one will be able to then match results of protein expression patterns and underlying DNA genotype.
Okay, Frederick, and another question is, what degree of correlation do you see between mRNA and protein expression?
That's an excellent question, and of course a question that everybody is asking. Very difficult to answer, mainly because the protein data is less secure and less quantifiable than the RNA data, which is then a more quantitative measure. What we can see when we do compare is that for the 5, 10, 15% of all genes which have some degree of tissue specificity, it relates a bit to the expression level on the RNA side. Saying that, I mean that if you have genes which are expressed in only single or very few tissues at relative high level of RNA, then the the correlation to protein is very good if one has a highly validated antibody. In general, my guess is that if you have a steady state population and a mixture as in a tissue, and one has a highly validated antibody, I would expect that the correlation between mRNA and protein is very high.
Okay, thank you. And another question. Why are there tissues expressing the protein AG, but RNA could not be detected?
Well, I would like to ask back, are there such cases or not? I mean, again, it relates to the former question. What about the antibody and if you have a highly reliable antibody, I would say that if there is no RNA, there should be no protein. That is very difficult to explain, although there are some things one could add to that the RNA data is not 100%. I mean, we know that there are transcripts with poly-A tails that we don't pick up, like histones, for instance. There you would expect a discrepancy between immunohistochemistry with a good antibody and your RNA data. I guess the only way I would say one more thing. It could be a technical artifact as well. And we are actually using the RNA data now to better validate our antibodies and also to perform titrations. So one could have cross-reactivity showing then a positive immunohistochemical signal, but if one then titrates the antibody based on the knowledge from the RNA expression, perhaps that staining goes away and you actually do see a very high correlation and no protein where there is no RNA. I think that's my comments on that.
Okay. Another question, person says, so you confirm a direct link between testes and brain based on the protein interactions?
No, not based on protein interactions. I mean, what we have done, and I think the question relates to the cytoscape plot showing that there are a number of genes, 46 genes actually, that are highly enriched in both the testes and the brain. It's interesting that if one looks at, for instance, geo-enrichment terms, there are no such common geo-enrichment terms for those 46 genes, different to the other pair of testes and lung, where 33 genes are highly expressed in only these two tissues, and those genes then they encode for proteins related to cilia function. But it depends on how you look at the data. I mean, if you look at correlation, the testis does not correlate very well to the brain, although testis and brain are similar in the sense that both these two tissues have the absolute highest number of tissue-specific genes expressed.
Okay, and another question. Nice talk, person says. Regarding the 216 tumors, 216 tumors representing 20 of the most common tumor types. That's an average of 10 samples per tumor type. Do you think that is a significant number to be able to make strong conclusions regarding protein expression in a specific tumor subtype? That was a mouthful.
Absolutely not. One should remember that this is screening, and as screening, I think 10 different individual cancers for each tumor type is quite a good screening number. What it does allow you to do is to see if a certain gene is differentially expressed within a cancer type or between different cancer types. But of course, it's only a starting point if you really want to draw any firm conclusions. So what you can see is you can conclude if a protein is expressed in all tumors or in just a few tumors. And if you see this differential expression, of course, your next step will be to build a larger cohort with a lot of clinical data and try to validate that differential expression and see if it has any bearing on patient outcome, etc.
Thank you very much. Henrik, question for you. For validation of antibodies, do you find it useful to block antibody staining with the engineered protein fragment that's used for immunization?
Thank you. That's, of course, a very relevant question, and from a historical perspective, a very relevant question, especially when it comes to using polyclonal sera. That said, in this case, all the AAA polyclonals have been produced and purified on the recombinant antigen. So in this case, it's not that important. However, we have tested it for a large number of our antibodies, and it works very well.
Okay. So here's a question, actually maybe two questions in one. The generation of antibodies to more challenging targets like GPCRs will most likely require alternative methods to those employed by HPR up to now. Can you briefly elaborate on how you are addressing this?
Thank you. Yes. Well, of course, it's a very good question. And of course, GPCRs are in some ways more challenging targets, perhaps, than perhaps other proteins. And especially the fact that often the loops are perhaps very short. That said, we use the same strategy in this case. We try to design our pressed antigens using really trying to find the regions that have the lowest sequence identity to all other parts on the human genome. However, in this case, the problem can sometimes be that the loops are too short, so we have to find alternative ways. One method we'll actually explore is to actually genetically fuse different loops together and to express them in one recombinant construct, in some cases with very good results.
Okay, thank you very much. And what about lot-to-lot variability in the polyclonal antibodies? How do you ensure consistency and ongoing availability?
Again, thank you for the question. I would say that reproducibility is, of course, a key feature for all antibody-based applications. And together with the Protein Atlas, we have done a tremendous amount of work, I would say, related to epitope mapping of the AAA polyclonals. And really try to compare different lots and different immunizations. And in general, I would say that using, in this case, peptide array mapping, we see 3 to 4 linear epitopes for approximately 100 amino acids. And these main epitopes tend to be found also in subsequent immunizations. And at Atlas Antibodies, of course, all new production lots are tested in parallel with existing lots to ensure functionality and assay reproducibility in a real lab situation.
Okay, and a question here, subcellular sublocalization. Do you have any information about mitochondrial localization or only nucleus and ER?
Yes, I think that's one of the beauties of the Human Protein Atlas. We're also trying to map the subcellular location of all the human proteins. And doing this, we use an ICC-IF approach using confocal-based microscopy. And we use a number of different reference markers to really show the nuclei, the ER, and the mitochondria. And we have been able to pinpoint up to 16 different localizations in the cell using the AAA polyclonals. And all this data you can find in the Human Protein Atlas if you go to the subcellular atlas.
Okay, well, thank you very much. Perfect. Hey, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we also highly recommend. A big thanks again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about topics brought up during the webinar. And thank you to our sponsor, Atlas Antibodies. Bye for now.